All right. Can somebody please confirm you can hear me?
Confirming.
Thank you. Okay, before we begin, are there any questions?
I have a question regarding the lab, if that's a possibility for us to talk about now, or if you want me to wait until the lab, then I can wait.
Is it a quick question? Because if it's not a quick, I want to wait till the lab.
It's mainly just on case study 5. I was just struggling with the 4 tests and knowing how to use it. So that is totally okay. I'll wait until lab.
Until the lab. All right, great. Um, I think I'm going to talk about the grading in the lab, so I'm not going to talk about it now. Let me go ahead and close down the grade book, and then I'll share my screen. Oh, come on. I've got too many Zoom things up. There we go. Oop. No, I don't have the main lab page open. All right, there we go. I had trouble opening Zoom for some reason. It wouldn't open initially. That's why I was a minute late. Um, so I had to restart it. Any questions before we begin, other than obviously about the lab? Let me share my screen here.
I have a quick question.
Uh-huh.
Do the lecture worksheets, like, would studying those, like, at all, like, help us at all with the chapter quizzes?
Yes.
Yeah? Okay.
Yeah. No, there's definitely questions about the worksheets on the quiz. Obviously, it's only a small part of the chapter, so it's not going to be a lot of questions, but there probably are questions from the worksheet on the quiz. Um, all right, let's go to the syllabus. So today is February 17th. Sorry, my voice is a bit hoarse. Um, we should be starting, actually, we should be on chapter 8. I'm not sure we started chapter 8 or not. And there is going to be a quiz 4 this Wednesday, so tomorrow. Quiz 4 will be covering Lab 7, Chapter 5, and Chapter 6. And note it will not be covering Chapter 8. Excuse me, not Chapter 8. It will not be covering Lab 8. Lab 8 was added late after this course was already developed. I think it was actually 2 terms after this course was developed. And so questions for Lab 8 will only be on the final. They will not be on any of the quizzes. Any question about any of that? All right, did we finish chapter 6? Can anyone?
I think we were close to the end, but I don't think we finished it.
All right, let me see where I am. No, that's not right. Sorry, they moved this around, the editing button. Get that out of the way. Well, I know we went past this. I'm sure we went past that. See if there's another one.
We were talking about the poisonous forms of oxygen gas.
That's the last thing we talked about?
I'm pretty sure because I filled out almost the whole lecture worksheet
Mhm.
last week. So
All
based
right,
off,
so we're behind schedule. All right, let me go look for that. I'm not finding, am I in the right chapter? Yes, I am. All right, have we talked about the effects of oxygen on cells?
Yeah, I think we were talking about that.
Have I talked about the different types of culture media? Let me take a look at chapter 8. I'm having a hard time finding out where I was. Um, Normally I put a link in there where we left off. No, there's no mark in there. All right. Can somebody give me again where we left off on chapter 6? And I'll try and find that.
I'm looking right now at your YouTube video to see what the last slide was that we covered.
Guess I can do that too.
Okay, it was the effect of oxygen on microbial growth to here. That was the table 6.1. That was the last slide that we covered.
All right. Thank you. So that's the last place we talked about. This slide here. Is that right?
Yes.
All right.
Yes.
All right. So let's talk a little bit about culture media. Media is a word for plural. So if you're talking about more than one medium, you use the word media. If you're talking about one medium, you use the word medium. Is that clear? A medium is a liquid or a solid solution containing all the nutrients needed for microbial growth. The media in microbiology is normally sterile. We sterilize it by putting it in the autoclave. So sterile media is one that has no living microbes in it. The inoculum is the microbes introduced into the medium. So you inoculate the media with a culture. A culture is the microbes growing in or on a culture medium. And we have a wide variety of media available in microbiology. If you were to come into the lab, I could show you a book that is about this thick, about this long. It's actually a little longer and a little wider. On in each page, we have one media. It's a recipe for making one media. So that's a large number of pages in this book, and that tells you the entire number of media we have available in microbiology. Any question about any of that? So a large number of media available for growing microbes and microbiology. A culture media is something to support microbial growth. A medium must provide an energy source, a carbon source, sources of nitrogen, sulfur, phosphorus, trace elements, although you usually get that from simply distilled water, or if you're putting in ground up cow parts, the trace elements come from the ground up cow, and or any organic growth factors. These have to be supplied in the media if the organism needs them. Now, if you're doing something like growing an autotroph that can pull nitrogen down from the air and can use its energy source for sunlight, assuming that you're putting in light to grow, what do you need to supply? Do you need to supply an energy source? It's a yes or no.
No.
No, because it can use the energy from the sunlight. Do you need to supply a carbon source in the media?
No.
No, because it's an autotroph that can use CO2 and it pulls nitrogen down from the air, so you don't need to supply nitrogen. You just need to supply sulfur, phosphorus, and then the trace elements, which usually come if you're using tap water. You'll supply trace elements by using tap water. To supply trace elements, you would not want to use distilled water. Any question about any of that? All right. And an autotroph isn't going to need an organic growth factors. Agar is a complex polysaccharide derived from seaweed. It's used as a solidifying agent to make a liquid culture media, a solid media. And there are other things we can put in the media to the liquid media to make it a solid, like you could put in gelatin or you could put in starch. But the number one is agar. Any question about that? Agar is usually used in a petri dish, but it can be used in a tube as well. A liquid media is usually put in a tube because if you put it in a petri dish, it's too easy to spill. Let me shut my door here. There's a bunch of noise coming from outside. Any question about agar and liquid media? A liquid media is usually called a broth. We usually put liquid media in tubes, a test tube, because if you put it in a dish, it's very easily to spill. We have two types of tubes. Let me get a paint up here. There it is. Took me a while to find it. Now, obviously, if you have a liquid broth in a tube, you only have a tube and then the liquid comes across. You can have a solid media in a tube, and if it comes across like that, minus this right here, this is called a deep. If, on the other hand, you have a tube like this, and then you use the auger and you put it at a slant like that, we call this a slant, which that's sort of supposed to come this way. And what you do is you have the tube and then you put it at an angle, and then at the angle, this is going across straight like that. And you allow the auger to cool, and then it'll harden in the slant, and then you put it upright. Any question about any of that?
Is there a reason we want to see a slant versus just a straight line across? Is it just to like extend the growth opportunities, like the surface area
Yeah,
for it?
if you want to grow your culture on a slant, there's more room to grow it on the slant than here. Um, I mean, it's even possible you can have something like that where it's first put in a deep and then you pour the slant on top, in which case it's like that. So you could have that too, but this is still called a slant. Um, Usually, if you're growing it in the auger, you put it in a deep and then you can stab it in. You could stab it in the slant as well, but it's better to put it in a deep if you're stabbing in the auger and then growing it inside the auger. They just have different uses. Agar is generally not metabolized by microbes, why it is a favorite in making the medium solid. If you were to use gelatin to make the media solid, there are a number of species which can hydrolyze the gelatin, and then you have a liquid mess. And actually, uh, who was it? Dr. Koch, when he first was using solid media, the very first thing he did was he cut a potato and then streaked it on a potato. I'm not sure if that was a cooked potato or a raw potato, but that's what he did initially. And then he started using gelatin in a petri dish. And the trouble with gelatin was that some of the microbes liquefied it, and then you have a gooey mess. And actually, it was the wife of one of his assistants who suggested using agar, and he tried it, and it worked so well, we still use agar today. Any question about any of that? We have different types of culture media. There can be a chemically defined media where the exact chemical composition is known. We know all the molecules that is in the media because everything is something you added. Let me show this up. So this is a simple complex media, a glucose salts media, which contains glucose, 5 grams of it, ammonium phosphate, 1 gram of it, sodium chloride, 5 grams of it, magnesium sulfate to supply magnesium and sulfur, 0.2 grams, potassium phosphate, mainly to provide phosphate, but also to provide potassium, 1 gram, and then you add that to 1 liter of water. There's no other molecules in this media. This is using distilled water, by the way. So that's what this is. E. coli can actually grow on this glucose salts media. Something like Streptococcus pyogenes, most members of Streptococcus cannot grow on this media. I'm not sure, but Micrococcus luteus, something that grows on your skin, I don't think it can grow on this one. But I don't remember. It's been too long since I've attempted. But definitely E. coli can grow on this media. Now, this is something that can work for a autotroph. All autotrophs would grow on that, as well as something that doesn't have many nutritional requirements. Something that's fastidious, where you need to add many growth factors, they won't grow on this. In fact, a fastidious organism will not grow on a general purpose media as well. And a fastidious organism would be something here. Let me see if I can blow that up. Bring that down over here. And it's a little bigger. So here you're adding just energy sources and carbon sources. And then you're just adding a bunch of ions. But here we have amino acids, cysteine, arginine, glutamic acid, methionine, asparaginine, a few others, cysteine. So if the organism requires these, it's fastidious, it will grow because we're adding it. And there we have B vitamin. There's another B vitamin. I think these two are B vitamins as well. And a fastidious organism may require many additives, like Streptococcus pyogenes. Actually, I'm not sure that one would even grow in this because I don't see nicotinamine adenine dinucleotide. I don't see that on here. So this wouldn't grow in that. But a fastidious organism is one where you have to add something, usually a growth factor, for the organism to grow. Chemically defined media are usually used for autotrophs because they can supply most of what they need. Any question about any of that? Another type of a media we call a complex media. It contains extracts and digests of yeast, meats, or plants, and these provide vitamins, organic growth factors, provide obviously a carbon source and a energy source. The exact chemical composition is not known in a complex media. Let me blow that up so you can see that better. Oh, that's interesting. I got two here there. Why do I have two here there? Oops. Go backwards. There we go. That shouldn't be there. Now I'll blow it up. Here is actually a nutrient agar. It contains peptone. Peptone is just a partially digested protein obtained from soybeans. Beef extract, 3 grams of ground up cow. Sodium chloride, 8 grams. Agar, 15 grams, and 1 liter of water. And this is what nutrient agar is. Very simple media. But obviously, the exact molecules will depend on what the cow has been eating. And different cows will eat different things, and they'll have different molecules in them. And so all of the molecules in this media are not known, because who knows what molecules the cows are providing. Any question about any of that? You could say the same thing about the peptone, although that is purified protein from soybean, but you could get contaminants in it, and that could provide molecules as well. So that's what a complex media is. Complex media is usually used to grow heterotrophs because they require an organic carbon source and an energy source. And we can use that for heterotrophic bacteria and heterotrophic fungi as well. Any question about that? Complex media are usually general purpose media, and they will not grow a fastidious organism because you have not added the growth factor that the fastidious organism requires. Like Streptococcus pyogenes will not grow on nutrient agar. Any question about any of that? We also have anaerobic media. This is important for organisms which are obligate anaerobes. You cannot expose them to oxygen, and so you grow them anaerobic media, and that way the microorganism is never exposed to oxygen because oxygen will kill them. Anaerobic media are usually reducing media. This is a media that contains a chemical like sodium thioglycolate that combines with and removes oxygen. What's called the reducing media. There's a component in the media that will remove oxygen, and sodium thioglycolate is one component. And I think I mentioned this media to you before. When you first take it out of the autoclave, there's no oxygen in the tube, but the tube is left open, and so oxygen comes in from the air. And at the very top of the thioglycolate tube, it'll be full oxygen. As you go down the tube, you have less and less oxygen until you reach the bottom of the tube or near the bottom of the tube where there will be no oxygen. And when you heat the media and the autoclave, that will drive off any absorbed oxygen in the media. Any question about any of that? We also have selective media. This is media that halts or inhibits the growth of a particular group of microorganisms while not affecting the growth of other microbes. One example of a selective media is 7% sodium chloride. This media has 7% sodium chloride in it, why it's called 7% sodium chloride media, and that will prevent most microbes from growing. Most microbes grow at 2% sodium chloride or less. 7% sodium chloride is a high salt concentration, and so most microbes will not grow on it. So this will select for a salt-tolerant organism, like Staphylococcus. Any question about any of that? Staphylococcus aureus, if you do not recall, is a human pathogen which can grow on your skin, and so it can grow on 7% sodium chloride. In fact, it can grow up to 20% sodium chloride, which is the highest your skin can get, depending on when you last had a shower or a bath, and depending on how much you're sweating. There are also differential media. This is media that has chemicals added to it that allow you to detect differences between two different species. So you have two different groups of organisms. If they grow differently on the differential media, they will usually view, meaning you'll see an observable difference. The difference is not related to how well the organisms grow on the media, but the visible difference make it easy to distinguish colonies of different microbes. One example of a differential media is blood agar. It's differential on hemolysis. You can have gamma hemolysis, that's the lower Greek letter for gamma right there. And you can see that the red blood cell is red all the way up to where the microorganisms are. Let me blow the slide up a little. Gamma hemolysis also means no hemolysis. You also have alpha hemolysis. In the alpha here, in the off-white color, that's the organisms. You can see the red blood cells are red up until a thin area of darkening around the cells. And inside the alpha, it's totally dark. That darkening is from alpha hemolysis, which is partial hemolysis of the red blood cells. Sometimes alpha hemolysis, instead of looking dark, it looks greenish. Just depends on the light and how you're viewing it. Here it looks dark. And that's different from beta hemolysis. Here we have in the beta, in the white, in the beta is the cells. Around the cells, we have a clear zone. That clear zone is because all of the red blood cells have been lysed. You can actually see through the clear zone of the media. So what I do is I like to hold it up to a clock or the light and then look through the media. And if I can see through it, I know that's beta hemolysis. Any question about any of that? Anyways, this is a differential media because you can detect differences between different forms of hemolysis, either gamma hemolysis, that's no hemolysis, alpha hemolysis, and beta hemolysis. And that makes it easy to distinguish between the different microbes. I'll shrink that down a bit. Now, you should realize that we have different types of culture media, like we have selective and differential media. Let me go back right here. This is a differential media, but it is also a enriched media, meaning one which has additives to it coming from the blood, making this a media that you can grow fastidious organisms on. So it's both a differential media and an enriched media. You can also call that an enriched general purpose media. Here is a selective and differential media, and it's EMB or eosine methylene blue. It's selective against gram positives. So if the organism can grow on the plate, it's likely to be a gram negative organism. It's differential on the basis of lactose fermentation. If the organism cannot ferment lactose, the organism will be its normal color. And this one, you can sort of see it. It is whitish in color, the colonies. This one can ferment lactose, and you can see it's dark in color, or right there, the colonies are black in color. Sometimes it might appear metallic green, but here it's looking black or dark. So it's selective that if there's growth, it's likely to be gram negative because it selects against gram positives. And if it's dark, it is fermenting the sugar, lactose. And if it's white, it's not fermenting the sugar lactose. Any question about any of that? Shrink that down. We also have an enrichment media. Don't get that confused with an enriched, ending in a D, enriched media. Blood agar is an enriched media. An enrichment media is one that encourages the growth of certain cells or certain species and discourages the growth of other species. So it is selective. An enrichment media is always selective, but we use it for a different purpose. An enrichment media is one that you use to enrich for microorganisms that you're enriching for, meaning, let's say you want to find a phenol degrading media from the soil. There are phenol degrading media cells in the soil, but there will be millions, at least thousands, hundreds of thousands of different species in the soil. And so you can put that on an enrichment media, encouraging the growth of phenol degrading microbes. And then that will increase the probability of phenol enriched media after you've grown it in the enrichment media. An enrichment media is particularly useful if the targeted microorganism is only a small proportion of the total population. It is often used for assaying soil microbes, other environmental microbes, or when you're checking for a human pathogen from a fecal sample. There's lots of microbes in the fecal sample, and so you may put it in an enrichment media to select for the potential human pathogen. Any question about any of that? An example of an enrichment media, as I've already mentioned, we're enriching for phenol degrading microbes in the soil. One possible enrichment media is a media where the only carbon and energy source is the chemical phenol. That will enrich for organisms that can degrade phenol because phenol is the only carbon and energy source. Other microbes that cannot degrade phenol cannot grow in the enrichment media. And what you do is you just have your enrichment media and you add microbes from the soil to it. And let's say it enriches 100 times, that won't be enough to be able to streak out and find your microbe. Let's say that your microbe is something like 1 in a million. But if you put it in the enrichment media and it increases 100-fold, it's going to be 1 times 10 to the fourth, which if you were to streak that out, you're not going to be able to find a colony of interest because the most you would have would be, well, 10 to 100. 100 would be a very high number streaking out. And you need 10 to the fourth colonies to get one, but you could put it in that another tube of enrichment media, and assuming that it enriches 100-fold again, it will drop from 10 to the fourth to 10 to the 2. And that means if you were to streak out 100 colonies, you could find one colony of interest, which is doable if you're to streak out. Any question about any of that? All right, this table is showing you the different types of media. We have chemically defined media, a complex media. We have reducing media for growing obligate aerobes. We have selective media to select against something we do not want and select for something we do want. We have differential media that allows you to differentiate between two different species, usually by the way they appear on the dish. And then we have an enrichment media, which is similar to a selective media, but designed to increase the numbers of desired microbes to detectable levels. Obtaining a pure culture is something you need to do in microbiology, as well as if ever you do work with microbes, you'll probably need to do that. A pure culture is one that contains only one species of cell in the culture. A colony is a population of cells arising from a single cell or one spore or a group of cells, like two or more cells clumped together. When we talk about colonies, we often talk about the colony forming units per mil rather than the number of cells per mil, because we don't know if the colony was formed by one cell, by two cells, or by a clump of cells. And so when you're counting the number of plates, and then you're extrapolating to the number of cells in the sample that gave rise to the colonies, you don't say the number of cells per mil, you say the colony forming units per mil, because you don't know if the colonies form from one cell, two cells, or a clump of cells. Anyways, you can obtain single colonies, which is a critical part for obtaining a pure culture. How you do that is streaking for colony isolation, also called the streak plate method. I usually call it streaking for colony isolation, where you have a mixed culture of cells. Let's see if I can blow this up. And then you streak it out on a sector or a quadrant, and then sterilize your loop, streak it out on another sector, and you sterilize the loop, and then you start streaking and you go through part of sector 1 where the sector 1 has been diluted. You don't go into the high concentration of sector 1, this region here. You go where it's diluted. And you should only go 3 or 4 times. This is a little too many here. 1, 2, 3, 4, 5 times, or maybe 10 times if you count that 1, 2, because you do go through that one twice. Like I said, you want to go through 3 or 4 times into sector 2. And the purpose of going into sector 1 is to pick up cells in the dilute region of sector 1 to bring them into sector 2. And then you streak out in sector 2 20 to 30 times. If you need to, you can repeat where you've streaked. And then you sterilize your loop, and then you cool it, and then start in sector 3, which isn't shown here, but you're starting here, and then you're moving there. And you shouldn't do it one time. You should go in 3 times, 3 or 4 times. So go into sector 2 where it's dilute. It shouldn't be right there, actually. It should be where it's more dilute in this region here. And then streak out sector 3. The purpose of this streaking for colony isolation and sterilizing the loop in between sectors, and then starting with no cells on the loop, and then going into the previous sector, is to pick up cells at the diluted portion of the sector and then bring them into sector 3, and then streak it out 20 to 30 times, not all shown here. And then hopefully someplace along the line, you'll be streaking out individual cells, which will then grow up to be isolated colonies. In this case, it happened in sector 3, but it doesn't matter which sector it happens. It could happen in sector 1 or sector 2. If the cells per mil was dilute enough, it could happen in sector 1, where you're putting out single cells, and you could get isolated colonies in sector 1. Usually, though, it's later, because usually you're using from a stock culture, in which case you have lots of cells on the loop. And as you can see, it's growing totally confluent in this region. And it's not even apparent that that's diluted from there. But if you look at sector 2, let me blow this up a little. You can see this is actually done correctly, that this part is more dilute than this part. In fact, in the end of sector 2, we're actually getting out individual cells being put down. Any question about any of that? All right. When you pull out a colony from here, you would think that that would be a pure culture, but in reality, 30% of these colonies are not pure. And that's because this colony, you don't know if it's formed by one cell, two cells, or a clump of cells. Now here we can see a visible difference between that colony and that colony. So it's likely that that is pure, but let's suppose this colony was formed by, I don't know, let's say 5 red cells and only 1 yellow cell, and the yellow cell grows slower than the red cell. That could be a mixed culture, and you're just not seeing the yellow in it because it's at low numbers. And so what you want to do to make sure you have a pure culture is take from an isolated colony on your first plate and then streak it out again on a second plate, streaking it out for colony isolation. If you get all of the colonies the same, then you know you have a pure culture. But let's suppose that this was from this colony here, and it did have a few yellow colonies in it, meaning a few yellow cells in it, and you see occasionally a yellow colony on sector 2 or 3. If you take from a single colony here that's isolated, it's gone through, the cells have gone through 2 colony isolation events. So if you pick from a second colony here and then set your stock culture up there, you're going to be 95% certain or greater that you have a pure culture. And the reason being is you took from an isolated colony here, and then you took your cells from an isolated colony here. So the cells have gone through 2 colony isolation events. Your chance of getting a pure culture is at least 95%, if not higher. Any question about any of that? Now, if that's stated in microbiology and actually all of biology, you can never be 100% certain that you have a pure culture because you never know if this colony was formed by 1 cell, 2 cells, or a clump of cells. And if it was formed by a clump of cells, you could have 1 yellow cell in it. And then let's say you pull from another colony in this plate, but let's say it was formed by a clump of cells as well. There could be 1 yellow cell in that clump, in which case this colony may be not pure. And like I said, in microbiology, you can never be 100% certain that you have a pure culture, at least by streaking out in colony isolation. I should word it that way. You can't be 100% certain just by streaking out, but you can be 95% certain or higher, and that's good enough to use in microbiology. And so when we say we have a pure culture, what we're doing is we're saying that we sent these cells through 2 colony isolation events and that we're certain that we have a pure culture at least 95% of the time. Any question about any of that? All right, the colony streaking method works well when the cells to be obtained, meaning purified into a pure culture, are large in numbers of the population of total microbial cells. If they are not, you then need to grow your cells on an enrichment media to increase the frequency of the cells of interest. Any question about any of that? All right, let me remind you, microbial growth refers to the increase in the number of cells, not in the cell size. When we're talking about microbial growth, you should realize that most bacteria grow by binary fission, where one cell splits down the middle literally and divides into two. So most bacteria grow by binary fission, but some bacteria can grow by budding, where one cell forms a small outgrowth called the bud that enlarges and separates from the parent cell when the bud has obtained about the same size as the parent cell. Any question about any of that? Some bacteria species, just a few, also reproduce by reproductive spores, where a spore forms at one end of a size, usually the tip of a filament, and then scatter. Let me draw a picture of that. Usually we have a parent cell and it'll put itself like that. And then it'll put the reproductive spores on this end here. I'm not drawing that. Why am I drawing that? There we go. All right. It's usually on one end of the cell, like the upper end of the cell, so that the wind or an animal coming by can pick up one of these reproductive spores and then take it elsewhere so it can be deposited and then grow someplace else. It'll eventually grow into a parent cell itself and then make reproductive spores. A reproductive spore is a form of reproduction because one parent cell can make 4 or more reproductive spores. And so one cell then becomes 5 cells, and that is reproduction. Any question about any of that? Remember, that's different from an endospore, where one parent cell makes one endospore, and then the parent cell dies. So one cell forms one cell. And that's why an endospore is not a reproductive structure, it is a survival structure. Any question about any of that? All right, let's talk a little bit more about binary fission. In binary fission, the DNA will first make a copy of itself and then split into two, and then one will move to one side of the cell, the other to the other side of the cell. The cell will then start dividing down the two, growing a cell membrane and a cell wall in between the two nuclei, literally splitting the two cell in two. And then the cells will finish their cell wall and then divide into two. Here we can actually see an actual electron microscopic image of a cell in the process of dividing into two, where there you can see the partially formed cell wall splitting the cell in two, and that's a nucleoid region or in a nucleoid region, meaning one molecule of DNA, one molecule of DNA. Any question about binary fission? The way that most bacteria reproduce. Some bacteria do reproduce by budding. I'm going to show you a little video of yeast budding. It's very similar to bacteria, where the parent cell puts out a little bud which goes on to form a cell. Not sure why I had the skip verification link. There it is. So here we're going to start forming a bud right here. And this is actually happening in a live cell. There, it's forming the bud now. It's dividing its DNA. And right there, you can sort of see the DNA moving into the bud. The bud now is about the same size as the parent cell, and you can see that the cell wall is formed. It's now isolated. And then both cells start to make a bud right here or right there. Any question about any of that? Anyways, that's how budding can happen. Microbiologists, when they talk about the generation time and graph it, they like to graph it on a semi-log plot, where on the x-axis, the generations is arithmetic scale, but the number of cells is a log scale. The reason for doing this is actually threefold, actually. First, when you plot the line, the cells form a linear relationship, y equals mx plus b, a very simple relationship. So it's easy to determine if you have this many cells at this time, how many cells you have later, because it's just a linear relationship, y equals mx plus b. On the other hand, if you were to use an arithmetic scale for the number of cells and an arithmetic scale for the generations, which is the same as the log scale here, semi-log plot, you'll have this type of a curve. This is not a simple solution. And the equation for this is something like y equals mx plus b plus nx squared and maybe plus bx to get this equation. Much more difficult one to do. Another reason for using a semi-log plot is that when you use an arithmetic plot, you can see that one cell divides into two, two into four, four into eight, eight into 16, and then 16 into 32, meaning we have a whole bunch of points all clumped together right here. If you use a semi-log plot, the points are more evenly dispersed. They're not all bunched right here. And then the last reason is that this one here shows you the last point we can get on the graph. The next point, we'd have to extend the graph up to the ceiling. Any question about any of that? On this one, you need to extend the graph to get the next point. Oops, should have gone there, sorry. So it should be on the ceiling here. But actually, it's going to be about right here. So you only need to extend the graph to right here. And then you can put this point here on the graph. You can't put this point on the graph because the graph doesn't go up to the ceiling. Any question about any of that? Anyways, that's three reasons why microbiologists like to use a semi-log plot. You can extend the graph easily and get the next point on. You have the points here separated out much better than you do here. And then the third reason is it's a much simpler equation. Any question about why we use a semi-log graph or plot to
No, but I
explain the
think you
generation time?
no, but I think you skipped a slide because we were just on the yeast budding.
Oh, right, I did skip generation time. The generation time is just the time required for a cell to divide from one cell into two cells, and it's the same number as the population number doubling in size, meaning going from 2,000 cells to 4,000 cells. They'll be the same time as the time for one cell to divide by into two. The generation time can vary between different species. It can be as fast as 20 minutes per generation, which would be E. coli. It can grow as fast as 20 minutes per generation. But most bacteria take 1 to 3 hours of generation time for the cell to divide into two and then go through another division. However, Microbacterium leprae, the bacterium which causes leprosy, has a generation time of 10 days, the slowest generation time of a bacteria that we can frequently or commonly grow. And this because the Mycobacterium species have a lot of macolic acid in their cell wall, and it takes the bacteria a long time to make that macolic acid into the component of the cell wall, which is macolic acid. Any question about any of that? Remember, when we're talking about the generation time, that we mean the time per generation. It is not the number of generations per hour. It is the time per generation. And usually when we talk about the generation time, we just say that E. coli has a generation time of 20 minutes. But what we mean is the generation time is 20 minutes per generation. The per generation is understood. Any question about any of that? All right, I already talked about that slide. Let's talk about the phases of growth. Bacterial cells, when you start them in a culture, they almost always go through the same bacterial growth curve, where initially you start the population from another population. Let's say you're growing from a liquid media, and then you're switching it to a solid media. It'll start with a lag phase, followed by a log or exponential growth phase, followed by a stationary phase, then followed by a death or decline phase. And surprisingly, this curve is found for all microbes and all living organisms on the planet. We know this because somebody introduced deer to an island off the east coast of the United States, and they initially were in the lag phase, then they started the reproductive phase, then the population stabilized and they were a stationary phase, and then the deer population started dying, entering the death or decline phase. Any question about any of that? The reason why the deer were decreasing is simply because of the population of deer not having enough food to eat on the island. Any questions? All right. With bacteria taken from a liquid media and then grown in a solid media, the bacteria normally enter the lag phase first. In the lag phase, the bacteria are not actively dividing. What they are actively doing is making the molecules the cell needs to grow in the new media, like the enzymes, various metabolites that are needed, they're being made. And the cell cannot grow until after all of those molecules have been made, or at least being made. Once the lag phase ends, we then enter the exponential phase, And this is the phase where the cells grow rapidly. Why are they growing rapidly? Because initially there's low numbers of cells, lots of nutrients, lots of space. Cells don't have any competition, so they grow rapidly, and you call that the log phase of growth or the exponential growth. That exponential growth will eventually begin to slow because up here, there are nutrients around, but less so than here. And there is some free space around, but less so than here. And then here, there's no waste products around because we just put the microbes in the culture. But here, there will be high numbers of waste products around, and waste products will slow the growth. We then enter the stationary phase, and that's the phase where the population number stays about the same, not increasing, not decreasing. This happens because the number of cells being born equals the number of cells dying, meaning the death rate equals the growth rate. The population stabilizes. Eventually, though, the population will enter the death or decline phase. In the early death or decline phase, there's more cells dying than there are cells growing. Why? Simply because the amount of nutrients is accumulating, it's getting higher and higher. The resources around are getting slimmer and slimmer, harder to find. I had one other reason. Let me see if I can remember. Resources, nutrients, oh, and waste products. And the waste products are increasing. The more waste products around, the slower the growth. That's early in the death phase or decline phase. Late in the death phase, you have no cells being born. Cells are only dying. It depends on the species, how long the species can last in the death or decline phase. For something like E. coli, the cell cannot be in the death or decline phase for very long, and all of the cells will die, certainly within 2 weeks, maybe earlier, a lot of cells earlier, but I don't know about all the cells. But by 2 weeks, the culture of E. coli will be dead. For Staphylococcus aureus and other Staphylococcus species, the death rate will occur, but it will not happen as quickly. And that's because Staphylococcus species probably live in a high saline environment like the skin, and so they will lose less water and they can survive longer. And then the longest living cell that we've ever seen is actually an endospore. And I don't remember, I think it was like 40 million years. They pulled an endospore out of the Antarctic ice core, thawed it out, and then got it to germinate, forming a parent form that we might not have seen for a long time if it was 40 million years. Like I said, endospores can survive a long, long time in a poor environment. Any question about any of that? All right. We have different measurements for measuring microbial growth. There are a number of methods. Some of them are direct methods, others are indirect methods. The direct methods are the plate count, also called the viable plate count, the filtration method, the most probable number method, and then the direct microscopic count. For indirect methods, we have turbidity, metabolic activity, and then the dry weight. I've used all of these methods for measuring the number of cells in a culture, except for the most probable number. I've never used that method. For the viable plate counts, also called the plate counts, it's the most common method used in microbiology to obtain your original cell population. What it's going to measure is the number of viable cells because you're going to count colonies, and a colony can only form from a viable cell. If you have a dead cell, this method will not count the dead cells, only the living cells. It measures the number of viable cells. It can take 24 or more hours for the colonies to form in the incubator, and then the results are often reported as the colony forming units per mil of the original sample rather than colonies per mil. We say colony forming units because we do not know if a colony was formed by one cell, two cells, or a clump of cells. Any question about any of that? All right, to get a plate count, first you make a serial dilution of your original sample, and then you inoculate the agar plates from the various serial dilutions, including your undiluted. Your diluted tenfold, meaning you put 1 mil of medium with cells in it into 9 mils of media. That's a 1 to 10 dilution. You shake that up, take 1 mil out of that, and put it into another 9 mils of sterile media. That's your now 1 to 100th dilution. And then you keep going for as long as you want. For how many dilutions to make? Usually we stop around a million, 1/1,000,000. And then you take your serial dilution, each dilution, and then set it up on a plate using the pour plate or the spread plate technique. I'll talk about that in a minute here. So there's your serial dilution where you have 9 mils of media and you add 1 mil of your original solution, which contains the cells. You shake that up, take 1 mil of that and put it in 9 mils. That's the 1 to 100th dilution. Shake that up, take 1 mil, put it into 9 mils. That dilution is 1 to 10, by the way. And then you have the 1 to 1,000. Take from that, put it into the next tube. There you have 1 to 10,000. Do that again. You have 1 to 100,000. And then you take an aliquot out of each dilution to spread it on a plate. Like you can take 1 mil, spread it onto an agar plate, get it to grow here. And then you count the number of cells that form colonies. And you're assuming here one cell forms one colony. Just remember in your back of your head that 30% of the colonies will be formed by more than one cell. And then you put it on the plate. I'm going to only talk about the spread plate technique where you put either 1 mil, oops, wrong one, 1 mil on the plate and then spread it around, or you put 1/110th mil on the plate, and you need to spread that one around very well with the spreader. And then you incubate the plate, count the number of colonies, and you take it from each dilution, and count the number of colonies. Let me blow this up. Many of the plates will be too numerous to count. TNT, too numerous to count. That one's too numerous to count as well. But that one you can count, and that one you can count. You want to use the count from this cell, because if you take it from this cell and then plus or minus one colony, that'll be plus or minus 25%, because there's 4 cells here. Here we have, I don't know, 30 to 40 colonies, and you'll have plus or minus 1 colony, and the standard deviation for the variation here will be much less than here. Whereas this one, it's going to be plus or minus 25%, meaning the mean here is 4, but plus or minus 25 to get 5% or 95% confidence interval. Whereas here you go plus or minus 1, it's going to be plus or minus, what is 30? Something like 3.3%. Any question about any of that? So you always take the colony count from a plate that has between 20 and 200 colonies on it. You don't want to count the number of colonies on the plate if it's less than 20. And then to get your original concentration of cells, you take the number of cells, what is that, like 40, times the dilution, which is 1 over 10,000. So if it's 40 times 10,000, meaning the cells per mil will be around 40,000 cells per mil in the original sample. Did I go through that too quickly? I'm assuming you guys know math, but that might not be the case. You have, let's say 40 cells is probably 30 to 40. You multiply by the dilution, that's 1 over 10,000. Which you multiply by this count here times the inverse of that. And then you get the original count in the original inoculation. And you report this as colony forming units per mil. You don't report it as cells per mil. You discuss it as colony forming units per mil. The reason being is we don't know if that colony there is formed by 1 cell, 2 cells, or a clump of cells. Any question about any of that? All right, the filtration method is similar to the viable plate count method. The difference is we use a filter, the filtration method, if our number of cells is very dilute. Like let's say you're sampling the bacteria in the city's water supply and you run your tap, you're not going to get very many bacteria in the fresh water. So what you do is you send the water through a filter. The water can move through, but the bacteria are caught on the filter here. And then you put the filter over a plate of media, and then the cells are brought from the filter, and then the nutrients come from the agar. And then you can count the number of cells. Like here, it looks like maybe 300 right there, but in this region there, that's a little bit less than 20. Or let's say 20, and maybe let's say 200 here. And then the number of cells per mil will be the number of cells here times the volume you pass through the filter will be the colony forming units for the cells per mil in the original solution. Any question about any of that? All right, the direct microscopic count is where a measured volume of liquid is placed under a defined area on a gridded slide. This method does not work good for bacteria that are motile, meaning they're moving. It can work on dead cells or slowly moving cells. Doesn't work when you need a high cell concentration, but you could centrifuge them, increasing the number of cells per mil. You can also, incubation time is not required because you need that time for the colonies to grow and then be visible colonies on your filter, or if you're talking about the previous one, on the viable plate count. So you count your number of colonies, multiply by your dilution, or divide by your dilution if you're sending cells through a filter. There's no incubation time because you've actually grown the cells up. So it does take, no, this is microscopic count. So it does take almost no time to do it, just the time of making a slide and then viewing it under the microscope. Here we're looking at a gridded slide. Here's the slide there. It's usually designed so that underneath this slide, underneath this square here, we have 1 microliter, and that means in the original solution, you need to take this number here, which is about 20, 15 to 20, and then multiply by 1,000,000. And then that will be your number of cells per mil in the original solution, which if this is 20 times 1,000,000 means it'll be something like 20 millions, or 2 times 10 to the 7th cells per mil. One disadvantage of the microscopic count in all other counts other than the viable plate method and the filtration is you cannot tell under the microscope, at least under the light microscope, is this cell living or is this cell dead? So the count will be the total number of cells. It will not be a viable cell. And the easiest way to get the numbers on the viable cell is to use the viable plate count technique. Any question about any of that? All right, that's
No,
for the
but just
direct methods. I won't talk about the most probable number, but we'll talk about the indirect methods: turbidity, metabolic activity, and dry weight.
just letting you know
Hey,
that we are at 6:22.
all right, thank you. Let me put a stop here. Are there any questions? Oops. All right, I'll see you in the lab at 6:30.
Professor, I do have one question for you. I sent you an email previously before class this morning, and I was just going to follow up with you to see if you received it or not. But I have
I
a
have seen some emails. I haven't had time to respond.
Okay, no worries. Well, I was just gonna request tomorrow. I'm having a back surgery earlier in the morning, and
Uh-huh.
so I'm gonna be out pretty much MIA for the full 24 hours that the test is gonna be available. And so I just wanted to see if it was possible if I could have the test due date for me pushed for Thursday instead, just so I could recover and not be taking the test all drugged up.
So you want to take it on Thursday rather than today?
Tomorrow the quiz.
Tomorrow's the quiz, yes.
Yes, so I was requesting if I could take it the day after.
All right, and you have an email requesting that? Because I won't remember this conversation.
Yes, I do. I have a message I sent you.
Okay, great.
Okay,
Oh,
so
yeah,
that
that'll
would
be
be okay?
fine. That'll be fine.
Okay, thank you. See you in
All
class.
right, bye.